So we want to look at the chemical pathology of the gastrointestinal tract. Chemical pathology of the gastrointestinal tract. So objectives: we will talk about anatomy and the physiology of the gastrointestinal tract, and causes, pathophysiological mechanisms, and diagnosis of the diseases. So we pay more emphasis on stomach, that is the ulcer, and pancreas. So we talk about acute pancreatitis and chronic pancreatitis. We also discuss malabsorption. We talk about small intestines, large intestines. Then we look at the causes, pathophysiological mechanisms, and diagnosis of vomiting and diarrhea. So a lot of topics. Let's proceed. So let's talk about anatomy and physiology of the GIT. Now you see, when you take the GIT, okay, you have this. Then we get to the stomach, okay. So this is the stomach. Can you see that one? Now the stomach is having some mucosal lining, okay. Remember that in the stomach we can get hydrochloric acid. The stomach walls produce the gastric juice. The gastric juice contains the hydrochloric acid. Some enzymes like the protease, of which we can talk about the renin, okay, the renin, which is found in the neonates like children, okay. You know, renin is very important because it converts milk protein caseinogen to casein for pepsin, which to the stomach. We talk about the stomach. Now from the stomach down there, in this portion is called a duodenum, okay, duodenum. Then you know the large intestine, the small intestines, okay. But in this video I'm going to pretty much emphasize on the stomach, the pancreas, which is here, the small intestine, and the large intestines, okay. So let's talk about them one after the other. So the stomach: storage and controlled release of ingested nutrients into the duodenum for absorption, okay. So when upon arrival of food substances, you know, protein foods are temporarily stored in the stomach. Some of them are digested in the stomach. So when you eat your beans, when you eat your meat, some of them are digested in the stomach. From there they move, they are temporarily stored in the stomach, and so they move from the stomach to the duodenum, okay. So storage and control release of ingested nutrients into the duodenum for absorption. You know, absorption doesn't occur in the duodenum, but from the duodenum goes to the small intestine for absorption, okay. Another one we talk about is initiation of protein digestion. So in the stomach there is protein digestion. So I spoke about a pepsin, which is a protein which breaks down solid protein food. It's an enzyme breaks down solid protein food, okay, convert them into peptides. And of course we have the hydrochloric acid also there. This hydrochloric acid plays a major role, okay. Sometimes if there are some bacteria in our food substances, it contributes in killing such bacteria. And so the stomach content is acidic, you know, but we talk much about the hydrochloric acid. Now the acid production to keep the upper GIT relatively sterile. So in the stomach there are some cells, for example the parietal cells. They ensure acid production, okay. Now what it does is that they produce acid to keep the upper GIT relatively sterile, you know. Yeah, so very important. Sometimes the acid production exceeds the normal range, and at least in some pathology of which we'll be talking about that. The next one I'll talk about is production of intrinsic factor for vitamin B12 absorption. Intrinsic factor is also a protein, you know, which is also produced by the parietal cells in the stomach, which is in absorption of vitamin B12, okay. You know, vitamin B12 is very important when it comes to absorption of folate and iron, you know. So if you like, you can get megaloblastic anemia there, you know. But in this video, that's not what we're talking about. We're talking about the function of the stomach. So these are basically some functions of the stomach, okay? Major functions. Always remember that there is production of hydrochloric acid there. There is production of intrinsic factor. So acid secretion by the stomach, I've spoken about that, okay? So ACL is produced by the stomach. Let's put it out. This is, always remember that ACL is produced, okay, by the stomach. Now in the stomach there are something we call the parietal cells, and it is the parietal cells that facilitate the production of the HCl, okay? It is very, very important. So after the production, it enters the stomach lumen, while the blood bicarbonate rises at the expense of chloride. So the HCl is produced by the stomach, and the production of the HCl, okay, occurs with some absorption of some ions, you know, some potassium channels and exchange of which I'm not going to talk about here. But always remember that it is the parietal cells in the stomach that facilitate the production of HCl. So the HCl production is stimulated by food or by stress, okay? Very important. There's a hormone I want to talk about. The secretion of a hormone we call gastrin, okay? Gastrin. Now gastrin also helps in the production of HCl, okay? Gastrin also helps in the production of HCl. So always remember that there's a hormone we call gastrin, which also facilitates the production of HCl. Very, very important there. So these two things, HCl is produced in the stomach by the parietal cells, and the hormone we call gastrin, okay, facilitates the production of the HCl, because gastrin acts on some mast cells and at the gastric mucosa to release histamine. Now what histamine does is that it acts via the histamine receptor on the parietal cell to stimulate HCl secretion. So in the presence of gastrin, there is a lot of HCl that is produced, okay? And then gastrin secretion is inhibited by acid-reaching G cells. So what I want to take from this slide is that gastrin facilitates the production of hydrochloric acid in the stomach, okay? Important. So this is talking about the, so there on the lumen there is parietal cells, okay? This is the parietal cell. There's histamine. So once gastrin acts on the histamine receptor, it induces the production of HCl by the parietal cells. A lot of process, but that is not our interest here. So intrinsic factor, we said, is also produced by the stomach, okay? It binds vitamin B12, which requires for absorption of vitamin B12. We spoke about that. So if you're deficient in intrinsic factor, it results in what is called pernicious anemia, where you are deficient in intrinsic factor so that you can't even absorb vitamin B12, okay? Sometimes it's accompanied by achorrhea. Achorrhea is lack of gastric HCl, okay? So renin, we also said stomach, there's production of renin there. So renin is a proteolytic enzyme produced in babies. What it does is that it clots ingested milk protein, casein, okay? Important. The pancreas is about 12 to 15 centimeters long and lies across the posterior wall of the stomach. It extends from the duodenum and spleen, okay? Now the pancreas has both endocrine function. Endocrine function is insulin. The pancreas helps in insulin activity or production of insulin, okay? Remember that the pancreatic B cells the activities in insulin production. So talking about the endocrine function, insulin, glucagon is also there, very important. And also have some exocrine functions, some hormonal functions, okay? So always remember that the pancreas has both endocrine and exocrine function, okay? So it secretes a watery-rich fluid which has a pH of 8, and its effect is to neutralize gastric acidity. So once there is acid in the stomach, the pancreas produces an alkaline watery fluid to neutralize the gastric acidity, so as to provide an optimal pH for pancreatic enzymes, okay? So that's a low volume viscid secretion of enzyme-rich fluid from acinar cells. Now some of the enzymes found in the pancreas, you have the amylase, the lipase, the ribonuclease, varieties of proteases. In fact, you can even get a trypsin, chymotrypsin, elastase, and carboxypeptidase. All these are produced by the pancreas. So always remember that the pancreas involved in the neutralization of gastric acidity, okay? The pancreas involves in the neutralization of gastric acidity. It is very, very important. So the small intestines, so when the food from the stomach, or chyme, the food from the stomach is called chyme. The chyme exits the stomach and enters the small intestine. The small intestine is made up of three parts: the duodenum, the jejunum, and the ileum, okay? So the luminal surface is composed of villi that are further divided into microvilli. Let's remember that that is actually where absorption occurs, okay? But then absorption of food substances occurs in the small intestine, let's put it that way. The large intestine starts with a circle separated from the ileum by the ileocecal valve, okay, anus. So here there is absorption of some water and some bicarbonate fluid there, Pathology of the stomach. This is where our interest is, okay? Pathology of the stomach, diseases, condition that can affect the stomach. First one we talk about is peptic ulcer, okay? Some people call it gastric ulcer or stomach ulcer, peptic ulcer. Now, in ulcer, there is a sore or a wound in the endothelial lining that protect the tissue or stomach walls from getting in touch with ACL. So I told you that the stomach produces hydrochloric acid, but this hydrochloric acid shouldn't get in touch with the stomach walls, okay? But what happens in ulcer is that there is a sore, there's a hole, or there's a wound in that endothelial lining, and when there is a sore there, its protective activity ceases, and therefore it exposes the stomach walls to ACL. So one may ask, what will cause the sore or the wound in the endothelial lining? Because at a normal circumstance, it's supposed to be there. There shouldn't be anything like inflammation or sore or wound. Okay, should be there to protect the tissue or the stomach wall from getting in touch with ACL. So in ulcer, what brings about a sore? So the major cause of the ulcer, the major cause of the sore is by bacteria, and that bacteria we call the Helicobacter pylori, H. pylori. This bacteria has the ability, H. pylori has the ability to weaken the mucosal lining, can weaken it. And once it weakens it, therefore exposes the endothelial lining to HCl. So there is an increased production of HCl due to the damage to the mucosal lining of the stomach walls, and that leads to ulcer. So always remember that in ulcer, the major bacteria here is Helicobacter pylori. Its activity is just to weaken the mucosal lining and expose it to ACL. So there will be a hole in the stomach. So we can see this from this diagram there. So you see this is the esophagus, the stomach. Okay, so you see that here there is a hole here. The endothelial lining of the stomach has been exposed to the ACL. So you can see this photograph. There's a hole there, and it results as a, from the influence of Helicobacter pylori. Helicobacter pylori. So always remember that Helicobacter pylori is a bacteria that induces the sore in the endothelial lining, leading to stomach ulcer or peptic ulcer. In the lab, this is what we do, of tests that we run. Okay, so the first one we talk about is the urea breath test. The urea breath test. What happens in the urea breath test? Here, the individual we suspect having the ulcer, okay, is given a labeled ulcer, is given a labeled urea. Consider a labeled urea. It is given orally through the mouth. Okay, through the mouth. Now, if H. pylori is present in the stomach, what is that? Is that H. pylori will spritz the urea via the bacterial urease. So the H. pylori is present. It will spritz the urea via this urease. Okay, the bacterial urease. Actually, it's an enzyme. Okay, so if H. pylori is present, it will spritz this labeled urea via this urease to spritz it to CO2, that is bicarbonate, which is absorbed into the bloodstream and exhaled as carbon dioxide. Okay, so to spread it to this CO2 move to the bloodstream and exhaled out as carbon dioxide. Okay, so it's a very nice test that they do. So somebody may ask, we have some other bacteria in the stomach, can't they also spread this? How sure are you that it is H. pylori? You understand that? But some of these bacteria, okay, are not, even though they are in the GIT, but they are not actually found in the stomach. You understand that? Bacterial activity is specific. Sometimes these bacteria that E. coli and those stuffs, they are not actually, even though they can even be in the stomach, alright, but they can't exhibit their activity. Okay, but here we are specific that to the H. pylori, which if this spreading of the labeled urea, okay, we are sure, 100% sure that it is H. pylori, and so this test is actually preferable as compared to other tests. Okay, so this one is very simple and it is very straightforward, and this is what most labs has engaged into. It's very nice test though. So we call it urea breath test. We always remember that a labeled urea is given orally to the patient. Once H. pylori is present, break it or split it into bicarbonate and ammonia. Now this bicarbonate here says CO2, right? It will move to the bloodstream, then to be exhaled out. Okay, then we are sure that H. pylori is present. Now when it's exhaled, we can measure in the lab. Okay, so they measure it and that. So preferred diagnostic modality for screening, diagnosis, and establishing eradication. Okay, this is the preferred one. Some antigen detection, they can detect H. pylori antigen is 2, but this one is not that specific. So we have the test strips, the cassette for H. pylori test for physics. Okay, you can also detect the H. pylori antigen. These two antigen can be measured by this immunoassay. And then we have serological test. So this one is very common in most hospitals, most facilities. Okay, so we call it H. pylori. You take your blood sample and drop it at buffer. If H. pylori, serological test measuring antibodies against H. pylori. This one sometimes, even if there has been treatment, the fragment of the antibodies may still be in the blood or the serum. So when it comes and it tests to be there, maybe there, or the antibody is not there, but still, the time period that it uses to clear from circulation. So this one is not that reliable. So the two tests, the antigen detection and the H. pylori test, they are not all that reliable. We preferred the urea breath test as compared to the two. Okay, we prefer the urea breath test to the two tests. Another condition that can affect the stomach is called gastrinoma. Okay, gastrinoma is actually a tumor. This tumor, remember I told you that gastrin is a hormone that facilitates the production of STL. Gastrinoma is actually a tumor which leads to excess gastrin secretion by the parietal cells. And what it does is that it causes what is called Zollinger-Ellison syndrome. The Zollinger-Ellison syndrome is basically a rare condition. UH tumors, which is gastrinoma, secrete large amount of gastrin, which causes the stomach to produce too much acid. Okay, so sometimes some tumors, like gastrinoma, can cause Zollinger-Ellison syndrome, where there is too much production of gastrin, which causes the stomach to produce too much acid. So in the lab, we determine basal acid outputs. So raised basal acid output in a setting of raised serum gastrin makes Zollinger-Ellison syndrome very likely, very likely. Okay, pernicious anemia and H. pylori infection should be excluded. So in pernicious anemia, we can get high amount of ACL, choroidea. In H. pylori too, there will be high amount of ACL. So we have to exclude all these two with a specific test. Maybe this one, you use the urea breath test. Pernicious anemia, there is a specific test for that, where you give the patient external intrinsic factor. So the bottom line is you exclude these two tests, and when there is still a basal acid output, a raised serum gastrin, okay, you suspect gastrinoma or Zollinger-Ellison syndrome. So essentially, a time sample of gastric juice is taken via nasogastric tube and titrated with sodium hydroxide to determine the amount of acid present in the result, and the result is calculated as millimole acid. Okay, so procedure involved in determining the basal acid output. Okay, very important.